A very good afternoon to all of you. Today we are going to discuss the biochemistry portion of Grand Test 183. Now let's switch over to first question. Which of the following is true regarding mitochondrial DNA? Now all of you know that mitochondrial DNA and nuclear DNA, there is a difference. Let's see the options. One set from each parents. Correct. It has 3 into 109 base pairs. It has more mutations than nuclear DNA. So the answer to this question is, it has more mutations. Now what is the reason behind this? The major difference is, if we talk about nuclear DNA, it has both introns as well as exons. And if we talk about mitochondrial DNA, since they lack this, obviously there are chances of more mutations in this. So the answer to this question will be C option. It has more mutation than nuclear DNA. Then next question. A child with pellagra-like symptoms, amino acids in urine, family history of 2 siblings affected and 2 normal, parents are normal, what is the diagnosis? Now pellagra-like symptom, all of you know, whenever we talk about pellagra-like symptom, it means what? The child is deficient in niacin. The precursor of niacin in the body is tryptophan. 60 milligram of tryptophan is required to generate 1 milligram of niacin. And that is why it is also known as a atypical vitamin, as it is synthesized from tryptophan in the body. Now the question is what? You can see child is showing pellagra-like symptom. That means the person is suffering from Hartnupp's disease. Hartnupp's disease, it is clear from the word itself, it was basically named after the discoverer. And Hartnupp's disease is followed by defective malabsorption of the tryptophan, which is the precursor of niacin in the body. So the answer to question number 225 is D, Hartnupp's disease. The next question, which of the following is an atypical vitamin? Just now I told you vitamin B3 or niacin is known as a atypical vitamin. The reason behind this is it is synthesized from tryptophan in the body. That is why it is known as atypical. Now see, the findings in the image is most likely to be associated with which nutritional dermatosis? The answer is B, scurvy. Now see, scurvy mein what happens is, there is defective synthesis of collagen. Collagen, which has a triple helical structure, it is synthesized from proline, lysine and glycine. The repeats of proline, glycine and lysine are present, and they are also present in their hydroxylated form. And hydroxylation of these 3 requires vitamin C. Deficiency of vitamin C will lead to defective hydroxylation of all the 3 amino acids, leading to scurvy. Then next question. Thiamine deficiency results in decreased energy production. Now see, thiamine, the active form of thiamine is TPP, thiamine pyrophosphate. This thiamine pyrophosphate is a very important coenzyme for the reaction enzyme PDH, pyruvate dehydrogenase, which is again an example of multi-enzyme complex. Alpha ketoglutarate dehydrogenase, which is a very important enzyme of citric acid cycle. And third is, it is required for transketolase activity. Transketolase, which is considered to be a very important enzyme of HMP shunt, in the non-oxidative phase. Now here the question is, thymine deficiency results in decreased energy production. Why? Because TPP is a cofactor of pyruvate and alpha ketoglutarate dehydrogenase. The reaction catalyzed by this is pyruvic acid will be converted to acetyl coenzyme A. The enzyme is PDH, which is an example of multi-enzyme complex as it requires so many cofactors and coenzymes to work. The most important coenzyme is thymine because it is actually taking out this carbon dioxide. This is a link reaction, or it is also known as oxidative decarboxylation. Decarboxylation means removal of carbon dioxide in the presence of oxygen. And obviously PDH, so NADH is also required. And why TPP plays a major role? Because it is the only coenzyme which is actually taking out this carbon dioxide in the first initial step. The rest are simple dehydrogenation reactions in which FAD is required and NAD is required, right? Then next, so the answer to this is it is a cofactor for alpha ketoglutarate dehydrogenase plus it is a cofactor for pyruvate dehydrogenase complex. Then let's switch over to next question. Nanotechnology has found tremendous application in the diagnosis of cancer because of the following advantages. Yes, now see, nanotechnology, study of the nanoparticles. Nanocrystals exhibit bright fluorescence, yes, it's correct. It has a narrow spectrum wavelength, yes. Peak spectrum wavelength is tunable, yes, we can adjust. And nanocrystals exhibit a narrow difference between their excitation and emission peak spectra. This is wrong. Nanocrystals always exhibit a very big difference between their excitation and emission peak spectra so that we can differentiate the particles. It has to be broad. Then only we can see the difference when it is excited and when the emission peak is observed. So the answer to this question is D. Then 230, graph given below is the titration curve of a biochemical compound. Which of the following statement is true? Now see, before taking this, you should know whenever we talk about titration of weak polyprotic acids or polyprotic compounds, two type of points are attained. One is the midpoint and other is the equivalent point. The ionizable groups are differentiated into their particular components. Midpoint is what? Midpoint can be defined as that point when the number of moles of the strong base, when the number of moles of a strong base equals to half of the weak acids which are already present. So midpoint is what? When the number of moles of a strong base, it will be equivalent to half of the weak acids which are already undergoing titration. And this equivalent point is what? Midpoint is what? It is half of the equivalent point. It is half of the equivalent point. When we talk about midpoint, obviously pH has to be equivalent to the pK whenever, and buffering strength is maximum at this point. Now let's look at the options. Options are what? You can see the graph here. See, there is a increase in pH here. Similarly, there is an increase in pH here also. So these are what? A and B points are representing the range of the ionization of the amino group as well as the carboxyl group. Because if you look at the other options also, you can see the third option is the compound has 3 ionizable side chains. As you can see, there are only 2 ionizable side chains, one representing the amino group and the other representing the carboxyl group. Fourth option is what? It has 1? No, it has 2. So the answer to this question would be B. The point A and B is representing the range of ionization between the amino group and the carboxyl group. Then question number 231, the sequence of compartment for gluconeogenesis. Now see, how do we define gluconeogenesis? Gluconeogenesis is formation of glucose from carbohydrate precursors as well as from non-carbohydrate precursors. And all of us know that gluconeogenesis is exactly a reversal of glycolysis. In glycolysis, the 3 steps which are irreversible, they are made reversible in gluconeogenesis. That is why we say gluconeogenesis and glycolysis are reciprocally regulated. If glycolysis is occurring, there is no question of gluconeogenesis and vice versa. Now see, what happens is, glycolysis is occurring in the cytosol. The resultant of glycolysis is that 1 molecule of glucose will be degraded to form 2 molecules of pyruvic acid. Pyruvic acid which has 3 carbon atoms. Now this pyruvic acid will be converted to acetyl coenzyme A, giving out 1 molecule of carbon dioxide by PDH enzyme in the mitochondria. Now, just now I told you the third step of glycolysis, that is conversion of PEP to pyruvic acid, would be the first step of gluconeogenesis, that means conversion of pyruvic acid to PEP. Now this pyruvic acid which is already there in the mitochondria, it will undergo carboxylation. This pyruvic acid will undergo carboxylation in the presence of enzyme pyruvate carboxylase, forming oxaloacetic acid. And a very interesting feature about this reaction is this acetyl coenzyme A is a positive allosteric modulator of this reaction. That is why we say gluconeogenesis is always accelerated during prolonged starvation. The reason behind this is acetyl coenzyme A, which is already coming from lipolysis, degradation of fatty acid, it is accelerating the conversion of pyruvic acid to oxaloacetic acid. Now what happens is, this is occurring in the mitochondria. This is TCA cycle. Now this oxaloacetic acid is impermeable to mitochondrial membrane. This will be converted to malic acid first. This malic acid is permeable. It will enter from the mitochondria to the cytoplasm, where it will again get converted into oxaloacetic acid, and this oxaloacetic acid will be finally converted to PEP in the presence of enzyme PEP carboxy kinase. So you can say gluconeogenesis is starting from the mitochondria involving the cytosol. Now what happens is, after the synthesis of PEP takes place, a reversal of glycolysis occurs. Reversal occurs, or you can say this was the last step of glycolysis, which becomes the first step of gluconeogenesis. Second step is what? Fructose 1,6 bisphosphate will be converted into fructose 6-phosphate. Simple, the enzyme is fructose 1,6 bisphosphatase. Now this fructose 6-phosphate will be converted into glucose 6-phosphate, and this glucose 6-phosphate will be converted into glucose. So you can say last reaction in which there is conversion of glucose 6-phosphate to glucose, this takes place in the endoplasmic reticulum. Enzyme is glucose 6-phosphatase. So we can say in gluconeogenesis, mitochondria to cytoplasm to endoplasmic reticulum. This is again a very important question. Gluconeogenesis involves 3 organelles. Mitochondria is where there is synthesis of oxaloacetic acid. Oxaloacetic acid, being impermeable to mitochondrial membrane, is converted to malic acid. This malic acid will be transported from the mitochondria to the cytoplasm, where it will be converted into PEP. Again, gluconeogenesis will occur, reversal of glycolysis is there. The last step, which is conversion of glucose 6-phosphate to glucose, takes place in the endoplasmic reticulum. So the answer to this question is B. Question number 232. Which vitamin causes toxicity? Now the vitamin which causes toxicity is, let's look at the options first. Vitamin B1, vitamin B2, vitamin B3 and vitamin C. Now out of these 4 vitamins, the vitamin which can cause toxicity is niacin, which is vitamin B3. This is question number 232. The vitamin responsible for causing toxicity is vitamin B3. And it is said also that if the dosage of vitamin B3 or niacin increases 500 grams per day, it leads to the damage to so many liver enzymes. Second important point is toxicity mein what happens is that the glycogen and fat reserves will decrease during niacin toxicity in the skeletal muscle and the cardiac muscle. Third important point about niacin toxicity is there will be increased concentration of glucose and increased concentration of uric acid. These are the 3 very important consequences of toxicity caused by niacin. First is there is depletion in the fat reserves as well as glycogen reserves in the skeletal muscle as well as the cardiac muscle. Second is there is increase in the concentration of glucose and hyperuricemia is there, and most of the liver enzymes are actually degraded, damage to the liver enzymes is there when the intake of niacin increases 500 gram in a day. And just now I mentioned this niacin being a very important vitamin, it is atypical as it is synthesized by tryptophan in our body. This is again a very important question. Precursor of niacin is tryptophan. So, and it is highly toxic in action. Then question number 233. Which of the following statements about nucleotide metabolism is not correct? Now all of you know nucleotides are what? Base plus sugar plus phosphate. Sugar is obviously deoxyribose in DNA and ribose in RNA. Phosphate, it is same whether it is DNA, whether it is RNA. So the basic difference between the nucleotide present in DNA and RNA are that DNA has thymine and RNA has, instead of thymine, uracil. And one very important fact about these is that these are synthesized by de novo pathway and salvage pathway. De novo means if the structure of purine is this and pyrimidine is this, the entire structure is synthesized within our body. The major difference is that purines are N9 glycosides, यहाँ पे they are attached to the sugar, whereas pyrimidines are N1 glycosides where they are attached to the sugar. You can say numbering always starts 1, 2, 3, 4, 5, 6, 7, 8 and 9. Numbering is anti-clockwise, and here it is 1, 2, 3, 4, 5 and 6, it is clockwise. Now see, de novo pathway is what? Which is occurring in all. Salvage pathway is what? It is single step reaction, because brain and RBCs, they basically lack a very important rate limiting enzyme of purine biosynthesis, which is glutamine amidinotransferase. That is why in brain and RBCs salvage pathway is operated, otherwise de novo pathway is operated. And in de novo pathway, the precursor of purines is ribose sugar. This ribose sugar forms phosphoribosyl pyrophosphate in the presence of enzyme PRPP synthetase, and this PRPP then forms 5-phosphoribosyl amine in the presence of enzyme glutamine amidinotransferase, which is actually absent in the brain and RBCs. It consists of 10 to 12 enzyme catalyzed reactions, and the major intermediate which is formed basically in purines is IMP. This IMP, inosine monophosphate, is finally degraded to form adenosine monophosphate and guanosine monophosphate. Or you can say purines are basically synthesized by taking IMP as one of the intermediate. Whereas in case of pyrimidines, simple. If we take the example of pyrimidines, CPS2 enzyme is required, which is a cytosolic enzyme. No IMP is produced in this. Only uracil monophosphate is produced, which forms thymine and which forms cytosine. This is how the synthesis of purines and pyrimidines takes place. Now let's look at the options. An early step in purine biosynthesis is formation of PRPP. Yes, I told you the enzyme is PRPP synthetase as it requires ATP. IMP is a precursor of both CTP and TMP. Now see, CTP and TMP are what? These are pyrimidines which are not synthesized taking IMP as one of the intermediate. Just now I explained, IMP forms AMP and GMP. Basically IMP is the one which is degraded to hypoxanthine. This hypoxanthine is degraded to xanthine, and finally uric acid is produced. So this is the answer. Third option is orotic acid is an intermediate. Yes, orotic acid is a very important intermediate of pyrimidine biosynthesis, and deficiency of orotate phosphoribosyl transferase leads to orotic aciduria. And fourth is ribonucleotide reductase converts ribonucleotide diphosphates. Yes, it's correct. A ribonucleotide reductase always converts the NTPs to DNTPs. So the answer to this question is B, IMP. Then question number 234. Deficiency causing manifestations shown in the image. Now see, it is clear from the image. There is the color of the tongue has changed, and you can see fissures around the corners of the mouth. So this is a typical case of deficiency of vitamin B2, which is riboflavin. Riboflavin or vitamin B2. Deficiency of riboflavin leads to glossitis, cheilosis, the color of the tongue changes, and Another important aspect I would like to mention here is in riboflavin, the anti-metabolite of riboflavin is galactoflavin. One important factor. Second important question regarding riboflavin is we have to carry out the assay of glutathione reductase in B2 deficient subjects. This is again a very important question. And RD of riboflavin is 1.2 to 1.7 grams in a day. Right? So the answer to this question is riboflavin. Next question is phenylbutyrate is used in urea cycle disorder. Now this is again a very important question. Phenylbutyrate, it is basically, it is also known as an orphan drug. Why? Because phenylbutyrate, orphan drug. It helps in scavenging this toxic nitrogen where urea cycle cannot occur, or it is bypassing urea cycle by actually reacting with this. Or what happens is this phenylbutyrate, it comes in the form of sodium phenylbutyrate. It can be taken orally also, right? Or it can be taken by nasogastric incubation also. Incubation. So basically what it is doing is it is combining with glutamic acid, because all of you know glutamic acid or glutamine. Glutamic acid is the major amino acid which is produced as a result of all the transamination reactions, and this glutamic acid finally undergoes transdeamination to remove this toxic ammonia, and it is converted to alpha ketoglutarate. Then this glutamic acid helps in the transportation of this toxic ammonia also by forming glutamine in the presence of glutamine synthetase. Basically what is happening, urea cycle is what? There is synthesis of this ammonia. The source of ammonia is glutamine, which is coming from glutamic acid. So what this phenylbutyrate is doing, it is combining with glutamate, and it forms those metabolites which can be excreted in urine with nitrogen present in them. So this phenylbutyrate is what? It is scavenging this nitrogen, right? Then next question is, a 3-month-old infant present with hepatosplenomegaly and failure to thrive, a liver biopsy reveals glycogen with an abnormal amylopectin-like structure. Now see, glycogen is what? If we see the structure of glycogen, see, glycogen has two type of linkages in it, alpha 1,4 and alpha 1,6. Synthesis of glycogen takes place in the presence of glycogen synthase enzyme. It's simple. What happens is, in glycogenesis, UDP glucose is there. This UDP glucose combines in the presence of glycogenin, which is the primer which is synthesized on the backbone of the proteins. It combines with this, and with the cumulative effect, you can say, of glycogen synthase, the entire structure of glycogen is synthesized. Now here what is mentioned is there is increase in the concentration of amylopectin, which is nothing but amylopectinosis, increased concentration of amylopectin. So obviously the deficient enzyme would be branching enzyme, isn't it? Branching enzyme which is catalyzing the branching which is incorporating alpha-1,6 linkage in the molecule of glycogen when in the presence of glycogen synthase and branching enzyme. So it's clear. The answer is branching enzyme. If you talk about alpha amylase, it has nothing to do with glycogenesis. Debranching enzyme is of glycogenolysis. Glycogen phosphorylase, it is the rate limiting enzyme of glycogenolysis. So the answer to this question is branching enzyme. Next question, number 237. Which of the following is correct about RBCs? Now see, let's look at the options. Deficiency of pyruvate kinase is not common. It's common. Glucose enters RBCs by sodium dependent? No, glucose always enters RBCs by facultative transport, and GLUT1 is the transporter which is required for the transport of glucose in RBCs. Third is deficiency of HMP pathway enzyme in RBCs will lead to anemia. Obviously, this is correct. What happens is, you know, HMP shunt, it is an alternative method of oxidation of glucose. Alternative method. It is a multicyclic process. It always starts with 3 molecules of glucose 6-phosphate. In this, ATP production does not take place. In this, CO2 is produced. And this is the major difference between HMP and EMP also. If we take EMP, which is nothing but glycolysis, Embedon-Meyerhoff-Pranas pathway, there you can see ATP is produced, but no CO2 is produced. So these are the 2 points of difference between HMP shunt and EMP shunt. Now the question here is, HMP mein there are 2 phases actually. One is the oxidative phase and second is the non-oxidative phase. First of all, you should know HMP is known as a shunt, not pathway. What is a shunt? Which does not have any end product. And that is what exactly happens here. Glycolysis is a pathway where the end product is pyruvic acid. HMP is a shunt because whatever intermediates are produced, they merges themselves into glycolysis. And what is it giving? It is giving NADPH. This NADPH is required for the maintenance of the cell membrane of RBCs because NADPH keeps glutathione reductase intact. Obviously, deficiency of NADPH leads to, there will be what? This is actually a cycle. What happens is, this glutathione is present in 2 forms. One is the oxidized form, other is the reduced form. To maintain glutathione in its reduced form, NADPH is required. And this glutathione, when it is in reduced form, it helps in the degradation or formation of hydrogen peroxide. So what is happening is, deficiency of this leads to splitting of peroxide free radicals. Free radicals. And all of you know, free radicals, they are in search of an electron. Whenever they search an electron, they immediately invade the membrane. And that is why deficiency of G6PDH, which is responsible for the synthesis of NADPH, leads to hemolytic anemia. So the answer to this question is, deficiency of HMP pathway enzymes in RBCs will lead to anemia. Excessive hemolysis will be there due to the release of the peroxide free radicals. And RBC does not contain RBC, does contain the enzyme of adenosine deaminase. Then next question, number 238. If aerobic glycolysis uses glycerol 3-phosphate shuttle, how many ATPs are produced? Now see, there are 2 types of shuttles. One is glycerol 3-phosphate shuttle, other is malate aspartate shuttle. Shuttle means it's like a shuttlecock. from one chord to another. So basically here the transfer of reducing equivalence is there in the shuttle. Most of the cycles are occurring in the cytoplasm, most of the cycles are occurring in the mitochondria. So the transport of NADH or the transport of FADH2, it's like shuttling from the cytoplasm to the mitochondria, because ultimately what is happening is mitochondria, since they are the powerhouses of the cells, everything has to enter into the mitochondria where they undergo electron transport chain forming metabolic water, and the amount of energy which is liberated at every step, it is used in the synthesis of ATP. Now let's see what do we mean by this glycerol 3-phosphate shuttle. Just now I told you it is a shuttle, so it is occurring between two organelles, cytoplasm and mitochondria. What is happening is this DHAP, dihydroxyacetone phosphate, it gets converted into glycerol 3-phosphate. Here the reducing equivalent is this glycerol 3-phosphate will be transported from the cytoplasm to the mitochondria where it is going to form FADH2 and DHAP will be transported here. So what you can see is कि glycerol phosphate shuttle में even though there is incorporation of NADH plus H positive, but in mitochondria FADH2 is liberating, because the question says in aerobic respiration. So we have to consider mitochondria, and this shuttle is like less efficient as compared to malate aspartate shuttle. Now what happens is you can see one molecule of FAD on hydrolysis gives 1.5. Since it requires two molecules, so 2 FAD will give 3. Plus what is happening, because it is coming from glycolysis, 4 ATP molecules are produced during substrate level phosphorylation and 2 are being consumed in the first and third step. So net consumption, yeah, net energetics will be 4 plus 3, 7 minus 2, that means 5 ATP. And that is what exactly happens. Aerobic respiration, which requires glycerol phosphate shuttle, it will generate 5 ATP molecules. So the answer to this question would be B, 5 ATP. Then last question, consider the following image, which of the following is most inappropriate. Now see, this is nothing but single mass spectrophotometer. What we are doing is we are studying the spectra of the proteins which is formed of peptides as well as the amino acids with the help of this spectrophotometer. What we do is, you can see S stands for the sample, where we are imparting the sample, and here the electromagnetic radiations are there. In the presence of this, obviously proteins या amino acids have zwitterine structure. They have amino group as well as they have carboxyl group. When in the presence of electromagnetic radiations, they are going to explode, and they will move by this pathway, and finally they will be detected by the detector present in it. This single mass spectrophotometry has basically overweight the Edmond degradation method because it is more sensitive. Now let's look at the options. See, it is more sensitive than the Edmond's technique. Yes, it is sensitive. Edmond degradation method is what? See, there were two methods actually, Sanger's method and Edmond degradation. They tried to study the sequence of the amino acids in a polypeptide. Since I told you that this single mass spectrophotometry is considered to be more sensitive because it is basically taking the aspect of both the things, mass as well as charged. Cannot detect covalent modification. This is false. It can detect covalent modifications. Just suppose if we do the mass spectrophotometry, what we do is, suppose we phosphorylate. Phosphorylation is addition of that phosphate. So what will happen is, suppose if this is the case, this spectrophotometer is considering the spectrum of all the compounds, whether it is phosphate, whether it is oxygen. So it is imparting what? It is going to impart phosphate is 1, phosphate has 32 molecular weight, oxygen is 3, 16 into 3, 48, it would be 80. So it is imparting 80 Dalton to it. So we are basically, you can say, separating them in a different phase. So it can detect. Electromagnet is an important component. Yes, electromagnet is an important, because everything, whatever is occurring, it has to be in the electromagnetic field. Then only there will be dispersion, ionization will be there. Sample chamber and vacuum pumps are important components. Yes, sample chamber is important, because unless and until we put a sample, nothing is going to happen. And vacuum is what? Vacuum is basically inserting that ionization in the presence of this electric field. So what happens is, there will be ionization, there will be movement of the amino acids as per their charges. And if we say higher is the molecular weight of a protein, higher will be the magnetic orientation which is detected by this detector. So the answer to this question is, they can detect covalent modification, right? So here we finish up with the discussion of the grand test 183. I hope all the questions are clear to you. Thank you so much.